All right, let's get the show on the road. Hi, welcome to the Physionic Podcast. My name is Nicholas Verhoeven. If you're not familiar with who I am, I'm a PhD student in molecular medicine, researcher at a medical school, and, well, I think that's all I'll talk about in terms of my credentials. Today we're going to be discussing, I think I always say this, but I'm going to go ahead and say it again, a really interesting paper that actually you guys voted on. So the Physionic community, specifically on Instagram, voted between two different studies to cover. And this particular study, which if you are watching this, I will throw it up on the screen, is called a Prolonged Fasting Drives a Program of Metabolic Inflammation in Human Adipose Tissue. Okay, so clearly, if you probably can pick out a few of those words, and it's telling you that we're going to be talking about how fasting, especially water fasting, it doesn't say that in the title, but water fasting affects our inflammatory process, or does it drive inflammation? And this is actually based off of not just this study, but this study actually references a few other studies that have also looked at and seen some effects of fasting on inflammation. So that's what we're going to be diving into. And there's going to be a lot of really, really interesting topics throughout this paper discussion. But I think that's where I'll leave it before I get too far ahead of myself. So like I said, the study investigated if water fasting led to a change in inflammation in men and women. So this is a human study, so we're not talking about necessarily in animals where you can do a lot more invasive work, which tells you more information in certain respects. And the researchers mention that caloric restriction, which is something I've spoken on before, as well as fasting in general, because fasting tends to lead to caloric restriction, leads to reductions in hypertension, atherosclerosis, which is that plaque buildup in the vessels, diabetes, and cancer. So kind of picking out some of the major ones, but certainly fasting has effects on all kinds of different other areas as well. But those are the big ones that affect at least the Western population. If you're talking about heart disease, you're talking about cancer, you're talking about diabetes, those are, that's a massive portion of the population that's under pathological strain. Okay, so, and they also then briefly introduce that fasting leads to increases in glycogenolysis. So a few different metabolic changes, or really physiological changes that occur. So specifically looking at glycogenolysis, which is the breakdown of glycogen. Glycogen is stored glucose. So stored glucose is just glucose molecules that have been stuck together into a chain, which eventually creates a network of glucose. And that gets stored in our liver and in our muscles primarily. Those are the two big sites. In terms of relative levels, the liver has the most, but in terms of total, like actual absolute number, obviously we have a lot more muscle than we do liver. So the muscle contains most of our glycogen stores, glucose stores. And fasting also drives gluconeogenesis, so the production of new glucose that's produced by the liver, and I believe the kidneys are also responsible for a small amount of gluconeogenesis. But usually we talk about the liver as the primary site. And ketone production. Again, if you're going to be converting fats into ketones for the brain, usually because of the brain, that also occurs in the liver. And all of this happens within about 2 to 3 days. And as we're going to see, fasting has a lot of different effects. We're not going to be so much focused on glycogenolysis and gluconeogenesis and whatnot, but we are going to be interested in one particular effect, which is called lipolysis. Lipolysis is simply the fancy term for saying a breakdown in fat. So you have fat that's stored in our fat cells and it's stored in other areas as well, but we're going to be focused on the fat cells. And when it needs to be liberated, when our body needs it, it will then shuttle those fat molecules out of the fat cells, which are two different things, and move them out, and that is called lipolysis. So once it is broken down, kind of like a storage unit, taken out of that storage unit, that is lipolysis. So the breakdown of fat molecules. So adipocytes, fat cells, undergo lipolysis by specifically dissociating. So it's stored as a triglycerol molecule or triglyceride molecule, which is a 4-part molecule. It is 1 part glycerol and it is 3 parts fatty acid. So 3 fatty acids are stuck onto a glycerol molecule, and that's what is stored in the adipocyte. Now, when the adipocyte wants to move that fat, it can't keep those pieces, those molecules stuck together. So what it does is it goes through 3 enzymatic reactions. It goes through an enzymatic reaction through adipose triglyceride lipase, hormone-sensitive lipase. That's probably the most famous one, if you want to call an enzyme famous, because it's usually used as a proxy measure of the other two in research, and monoacyl glycerol lipase. And that is the canonical pathway, meaning that is what is typically looked at. That's what you would consider when looking at fat loss or looking at lipolysis, the breakdown of those lipids. So you're breaking apart the glycerol, separating it from the 3 fatty acids. And really what we're going to be focused on is that whole process, but along with a really interesting secondary process that research is starting to become more and more familiar with. And I did not know this, so this paper was really interesting, and I learned quite a lot, and it's going to tell us what happens to these triglycerides aside from lipolysis, and it's also going to tell us a little bit about, does fasting increase inflammation? I'm going to go ahead and tell you the answer to one of them. Does fasting increase inflammation? The answer is yes. Okay, so we're going to go ahead with that idea moving forward. However, that does not mean that it is necessarily a negative thing. So we're going to go into that as well. So I'm going to give you a bit of background and then a little more background, specifically with images as well, if you're watching this. If you're not, don't worry about it. As always, if you're just listening to the podcast, I'm going to be explaining everything. And then I will move into kind of a breakdown of the storytelling, the data of how they got to these different conclusions, how the study was designed, how they were able to come to some of the conclusions, what we can actually take away, because I do think that they sort of stretched some of their data when it came to their presentation. But we'll talk about specifically what we can really take away. Okay, so if you're watching this, I just put up a graphic of, and I'm going to detail it for those that are just listening on the podcast. You have blood vessels that supply blood to different tissues. So in this situation, we're talking about fat tissue. So fat tissue has to be able to move fat molecules in and out of itself. So if the body needs fat molecules for metabolism or whatever it might be, cell membranes, all kinds of other things, fat isn't just for metabolism, then it will essentially call on these fats and release those fat molecules from the fat cells. And this process, obviously, like a highway network, works through these blood vessels. But within these blood vessels, you don't just have, fat molecules. You have red blood cells and you have white blood cells. But more specifically, when you're talking about immune cells, we have what are called monocytes. So monocytes are part of the innate immune system. They just go around in our bloodstream, and they're essentially just like sentries. They just go around from one area of the body to another area of the body, and they're just looking around, seeing what might be wrong. But inflammation, this is something I've been trying, and I need to do a better job of explaining this and really putting more content out about this, that inflammation, the word inflammation, people, for lack of better words, they bastardize it. they just use the word inflammation, but they don't actually give it proper context. And that is going to be so important as we go through this research, this particular science. So monocytes, which are part of the inflammatory process because they are immune cells, will actually then differentiate, meaning that they will essentially program themselves into another cell. And that other cell is a macrophage. So this macrophage will then infiltrate. I'm giving you an overview of what this paper is saying. This macrophage will infiltrate into the fat tissue. Once it enters the fat tissue, the fat cells are dumping fat so quickly. This really blows my mind. I really think this is super cool. So quickly that the macrophages are helping the fat cells by taking up the fat that is being dumped extremely quickly and helping in the breakdown process. So the fat cells aren't necessarily going through lipolysis, or maybe they are, but they're not able to release fat as quickly enough. So they end up employing these macrophages by releasing full triglyceride molecules. Remember, I explained to you that lipolysis does not use full triglyceride molecules. You start out with that, but you have to break up the glycerol from the fatty acids. Well, the fat cells are saying, we can't handle this. So they just start dumping it all. So they dump these entire glycerol molecules, or triglyceride molecules. And then the immune cells take them up and presumably then help by facilitating that conversion to create those fatty acids and then release and then use those for energy or for any other function of the cell. I just focus on energy because that's what a lot of people like to focus on, the burning of fat. Okay, so that is the overview. Now, we're going to go through the data after I go through a little bit of the study, like how exactly they went about probing these questions, and we're going to see if that truly is the case. And I will tell you from the get-go, there are a few things that I think they're stretching a little bit, but that doesn't mean that we can't take away quite a bit from this study. Okay, so study design. Let's talk a little bit about that. So the study design was set up in such a way that they had only 7 participants, and they don't need a whole lot of participants for this because a lot of the research that's actually being done is using the fat cells themselves. So if you take a biopsy from someone, which is usually done with a needle, so they'll stick a needle after they've numbed that area. They'll stick a needle into a person and take let's say 50 milligrams of something, if that's muscle tissue, if that's fat tissue, whatever it might be. And obviously some are more painful than others, but I think fat tissue is probably one of the least painful ones that you can do. And so they had 7 participants, men and women, although primarily women. I think they had 5 women and 2 men. They were overweight, but they were young, so 29 years of age. And they didn't have any other health issues from what I remember. I don't think they probed that too much, though. So they employed these 7 participants, had them come into a lab, so an incredibly controlled environment. And then they were instructed to only drink water plus a few like electrolytes or something, no calories though, for 10 days. And they were told to stay at that particular place. So while they're staying at this, it's usually called a metabolic ward, but I don't know if that's exactly what they ended up calling it. But regardless, it's a place that's incredibly controlled. So, I mean, they can leave whenever they want to, but they're supposed to for the study. They'd probably get excluded from the study if they decided to leave. So I'm sure these people are being paid a pretty hefty amount, like, I don't know, $500 or $1,000 or something like that to stay in this controlled environment. And they're told to fast for 10 days on water. So the researchers took a fat sample at the very beginning, so before the fasting really started, then 24 hours later, and then at the very end, the 10 days later. So that's all you need to know. That's all you need to know for the actual research design. And now we can actually go into what the researchers ended up finding out. They did a lot of transcriptome analysis, which is it's a fancy way of saying they looked at a lot of gene transcription. Let me see if I can break that down further. Genes are the DNA that's found within our cells. So the DNA can be read, and then that leads to a transcript. And you can almost think of it like the literal word transcript, like a written document almost. So a transcript of that DNA, which has certain information in it, and then that information is then read to produce proteins that then function within the cell. So you measure gene expression by looking at that transcript. How much of that transcript is there? And if a lot of it is there, then you can measure that. And if a little bit is there, you can measure that. So then that gives you at least a relative level of how much that gene is being expressed. So whenever I say that a gene is expressed, I mean that there's more of that transcript that's being read and being produced. Okay, so the first piece of information is something really basic, but I want to go over it anyway. Looking at, did the participants lose weight and fat? And hopefully these participants would lose weight and fat because they didn't eat anything for 10 days. They simply drank water. So if you're looking at the data, which I always repeat this, but really if you're on the podcast, I'm going to be explaining everything. You can see that clearly that, yes, they do lose weight. And I didn't show the body fat, but the same exact thing is true for body fat. So they saw drops in body fat. So great. In terms of kind of absolute numbers, they lost about 7 kilograms, so lost about a little over 15 pounds over a 10-day period. I mean, that's pretty substantial. But again, keep in mind that these people were overweight, so they had a lot more weight to lose than your typical person. So then the researchers wanted to know, were there any strange or kind of odd gene signatures? And this is why I explained the transcriptomics or transcriptome analyses, because this is where this kind of gene expression stuff starts to come in, and it can be a little overwhelming at times. So I'm trying to make sure that I'm keeping things as broken down as possible. So they're comparing the overall gene expression. So they took these fat cells from these individuals, so from these 7 individuals. They then plate them, so they put them on plates, and then they're able to keep them alive for a particular amount of time while they do experiments on them. And that's really where I'm going to keep that. I don't want to bog things down too much with the research science, the experimental research. So in this situation now, they're looking at the gene expression. Again, how much are these genes being expressed of particular genes? They did a comparison of from the first biopsy, which was when they weren't fasted, to the next day, 24 hours later, they took another biopsy, and then that 10-day mark where they took the final biopsy, and they looked at the gene expression of all 3 biopsies, kind of side by side. And they noticed that about 6%, I believe it was 6%, let me quick check my notes. Yes, 6% changed dramatically. 20% changed, but 6% changed dramatically. So that left about almost 1,600 genes changed really dramatically. So that makes up that 6%. So then from there, they were able to compare the genes from one time point to another time point to another time point. And if you compare the day 0, so at the very beginning, to the day 1, there isn't much change. If you're actually watching this, I've got kind of graphics and stuff kind of explaining things, but hopefully you understand, you're just supposed to look for kind of a general trend in this heat map, which is what I've got up. So you're just comparing the colors. If there's red, there's more gene expression. If it's blue, then there's less. gene expression. Simple as that. But where do you see a difference? That's really the big question. And where do you see a difference for those on the podcast is at the 10-day mark. At the 10-day mark, suddenly a lot of genes that were not expressed, that were turned off, suddenly become expressed. So, okay, that's really interesting. So clearly water fasting has some sort of change in these gene expression, gene signatures that they specifically looked at, those 6% of genes that they looked at. Now, without going too much more into detail with this, they did look at lipolysis genes, which is what I described earlier, right, with fat breakdown, the canonical pathway, the standard pathway that most people talk about. They also looked at the opposite, lipogenic genes, so the production of more fat. Obviously, in a water fasting situation, if you're losing body fat, do you think that your body wants to be then producing fat? Most likely not. But still, it's nice that they actually check this, just because then you know, okay, we're not lying to ourselves. We're not letting our bias dictate us. So at least they checked it. And there were decreases they did a bunch of gene measurements, again, gene expression measurements for lipogenic enzymes, because lipogenesis, de novo lipogenesis, is mediated by a series of different enzymes. So they just looked at those enzymes. Hey, is there more or is there less? And they found that there's a less, which makes sense. If you're water fasting, you're going to see decreases. But interestingly, here's where things get more interesting, is that with the lipolysis genes, some were increased and some were not. And then they did a little more digging, and they found that genes specific to the lysosomal pathway were increased. Now, if you're not familiar with what that is, the lysosomes are an integral part of autophagy. They're also an integral part of creating essentially vesicles to kind of trap molecules within them, and then to potentially release them out of the cell. So just to capture things and then push them back out. So they create these kind of bubbles, and inside the bubbles are a bunch of molecules. So that's obviously pretty interesting. I mean, the lipolysis genes were decreased, but the lysosomal pathway was increased. Strange. Okay, so but this at least gives the researchers some cause for pause, and suddenly they're like, okay, well, now let's investigate that a little further. So then they were curious if there was an alternative pathway for lipid breakdown that was afoot. So they decided to assay a particular gene, or test a particular gene in macrophages known as SPICE, S-P-I-C. So this particular gene, see, this is where they're kind of drawing back on the literature. So they've said, hey, other studies have found that there's kind of an inflammatory something going on, and the lysosomal pathway is certainly involved in inflammation. Not necessarily as a bad thing, it's just associated with it. So we're curious if, based off of previous literature, if we're seeing these changes that are somehow related in fat cells to macrophages, which are those immune cells that get converted from monocytes to macrophages as they move into that tissue. So just tissue in general. It doesn't have to be fat tissue. It can be muscle tissue even. Just as a fun fact, if you're lifting weights or if you do intense exercise, you do damage to your musculature sometimes. After a while, you become better and better and better, and your muscles become really resilient, so they do less and less damage. But at the very beginning, at least, when you're really sore, you're wrecked, right? You've got a bunch of macrophages, actually, that are going into that region and helping repair and fix that whole process, as well as sometimes becoming a little overzealous and doing their own damage. But that's a subject for another podcast. At any rate, they ended up measuring the gene expression of this SPICE gene. So again, if you're watching this, I have the graph up for you. But this spice gene is expressed when macrophages, again, those are immune cells, are stimulated. So what they did is they put macrophages onto a plate, and then they stimulated them with a variety of different things. So in the control condition, they didn't do anything. They added iron, which is labeled as hemin, and that has been known in other macrophages, more specific to bone, has been shown to increase their activity. So you would typically see an increase in spice. Now here's where you have a bit of a drawback in the paper, because you don't see that increase. If you're listening to this, I'm just describing a little bit of the data. You don't see an increase in the iron. So that would bring about questions of, well, okay, are you guys doing this correctly? Well, it turns out that they did other experiments as well. And yes, it seemed to have an effect, but for some reason in this situation, it didn't. However, then they also added two other things. They added, one, a saturated fat, and two, they added an unsaturated fat, so separately. So they added 50 micromolar or 500 micromolar, and you don't need to know what micromolar is. Just know it's an increase in concentration. So you're essentially adding more saturated fat. You're adding either 50 or you're adding 10 times more of that saturated fat, or 10 times more of that unsaturated fat. And what did they find? Well, they found that with the highest concentration of unsaturated fat, you see this greater expression of this spice gene. So in the macrophages, there was an increase in the spice gene. So then what they did is, and I'm not going to throw up the graph for this because it gets a little confusing, but they again wanted to look at the gene expression, but they didn't want to look at the gene expression in fat cells. So what they did is they did the gene expression in immune cells, so macrophages. And this is in a different line of macrophages. So these are not from the same participants that we've been talking about. So that is a limitation of this study. But they ended up looking at the gene expression that was just coming out of the macrophages, but you usually separate out the gene expression from M1 and M2. So M1 is a pro-inflammatory state of macrophages. So what they did was they expressed this spice gene, and I don't really know how to pronounce it, SPIC, however you would pronounce that. I'm just going to call it spice, even though it's missing the E. This spice gene. And then they want to know, okay, is there increases in that inflammation or in that inflammatory profile of the macrophages, wherein the macrophages will become more aggressive and will become pro-inflammatory. They start releasing other cytokines, meaning communication proteins, to other immune cells and other nearby cells to essentially recruit even more immune cells. And then they start kind of looking around and trying to figure out ways of battling whatever is triggering them to be active. So they looked at genes for that, but they also looked at M2, which is anti-inflammatory. So even though we're talking about inflammatory cells, that is not always the case. It's not always like you just look at an inflammatory cell like a macrophage and immediately say, okay, well, if there are macrophages there, then immediately that means that you have pro-inflammatory macrophages. That's not the case. And even this is a rudimentary system. I mean, even current research, M1 and M2 are kind of a simplistic way of looking at it. I mean, it's really on a gradation scale. So sometimes they're extremely M1, they're extremely active, like during an infection, for example, or if they're doing some sort of housekeeping activity, then they tend to be extremely M2, and sometimes they can be in the middle. So what happens when the researchers then overexpress or just express this spice gene? Well, they find that there is increased a series of genes. I think it was like 13 macrophage genes were highly expressed. And the spice expression led to this expression not just in M1, so pro-inflammatory, but also in M2. So we're getting kind of conflicting responses here at this point. So next I want to draw our attention, because technically there's one more piece of data, but I want to leave that for the end. Then they wanted to look at systemic inflammation. So systemic inflammation, the way they did this was just by looking at the blood levels of particular cytokines, inflammokines, or whatever you want to call them, immunomodulatory proteins. We're getting real complex here. But essentially, they're just cytokines. They're just communication proteins that either recruit more immune cells, or they try and push immune cells away. And they can either recruit those immune cells to be pro-inflammatory and aggressive and try and find some sort of problem to fix, or be anti-inflammatory, much like M1 and M2, like I was describing earlier. So what do they end up finding? So this is systemic. This is across the body. This is not just necessarily just in the fat cells or around the fat cells. So they looked at interleukin 10, tumor necrosis factor alpha, interleukin 6, and CCL2. CCL2 is a cytokine that allows for is a chemoattractant, so it attracts immune cells to a particular location. And so they're looking at 3 that are kind of pro-inflammatory. And I should mention here that just because I'm saying something is pro-inflammatory, that doesn't mean that marker is always pro-inflammatory. I mean, it's always context dependent. And that's why science, especially, well, in this case, I'm talking about biological sciences, they can be pretty tough to kind of tease out, because you really have to know the context and understand. I mean, we understand like what a cytokine does. We know what that particular cytokine does, but is it attaching to macrophages, and what's happening to those macrophages? There's a lot of things that you have to take into consideration. So they looked at interleukin 10, which is which is typically, I think that's the word I'll use. It's typically an anti-inflammatory cytokine. Then you have TNF alpha, which is typically a pro-inflammatory. IL-6 is pro-inflammatory, and CCL2 is considered pro-inflammatory as well. So, and what did they end up finding? Well, they found that interleukin 10 was significantly increased with water fasting. So after 10 days of water fasting, there was an increase in this IL-10. Tumor necrosis factor alpha, which is pro-inflammatory, was also significantly increased, and IL-6 was also significantly increased, but they did not see a difference in CCL2, which tells you, if you're a scientist, that you should usually try and have more than one measure of pro-inflammation, or really just in any situation. You typically want more than just one measure, just to make sure that you're getting the results that you're getting telling results. So what does this tell us? Well, it tells us that systemically, you're not getting this massive influx of pro-inflammatory cytokines. And that's why I was alluding to at the very beginning that we can't necessarily say that just because you have increases in immune cells, that suddenly you have increases in destructive inflammation. It's just a key point that I really need to mention. So it tells us that overall, there's an increase in these inflammatory measures, but it doesn't tell us that they're necessarily pro-inflammatory. Okay, now for the last bits of data, I wanted to discuss something that they didn't even leave to the very end, which I was a little surprised by because it just fit in nicely at the end. Then what they did is they went back to these participants, well, not to the people themselves, but more so to their samples, and with what samples they had left. They then wanted to see, okay, well, if we take slices of these biopsies, just, incredibly thin slices, and then we have someone else who doesn't know about the conditions, they don't know anything, but they are trained in identifying macrophages, will this person detect in images more macrophages in the adipose tissue? So is there going to be a greater infiltration of these macrophages with the water fasting condition? So they compared the no fasting, so at the very beginning, to 10 days later. And again, that's where we saw that massive change in gene regulation, which we did not see after 1 day of water fasting. That's why they're focusing on the 10-day compared to the baseline, to the no fasting condition. And what do they find? Well, interestingly, as you can probably imagine, they see increases in this macrophage infiltration, which is really, really interesting, because then that tells you that there is some sort of an immune modulatory impact of fasting. So these macrophages, based off of what I told you in the introduction, it does seem like there's some proof there that macrophages will then move into this tissue and just sit there. But then what are they doing there? That's really the next question. And while I will go ahead and say that this next piece of data is not a definitive explanation of exactly what's going on, it does at least make us interested. It makes us think, okay, this could be the mechanism. So they took images of macrophages, and then they stained these macrophages for lipid droplets. Lipid droplets are just vesicles with, well, lipids, fats in them, so triglycerides. And what they found is, sure enough, these macrophages that were in this adipose tissue, in this fat tissue, had higher levels of, or had these lipid droplets within them, which was odd. So now they're kind of bridging this gap between this initial idea that I was talking about at the very beginning, where you have these macrophages that are infiltrating into the adipose tissue, and as the adipose tissue is not releasing fatty acids, or not fast enough, it just dumps entire triglyceride molecules. Those get picked up by macrophages and then get broken down or get dealt with in some way, and we don't actually know what that is. We don't actually have proof, though, in this paper that the macrophages are doing anything with it. It could be unhealthy. It could be, I mean, all we know is that macrophages go into the adipose tissue with water fasting, and we know there's systemic inflammation. We also know that it's not pro-inflammatory necessarily, where the macrophages are then doing a bunch of damage. They might be going in there and just kind of dealing with whatever environment is there, and it may be a high lipid environment that, as the fat cells are releasing this fat, so the macrophages are picking it all up. And maybe they're breaking it down, at least, but that is speculation based off of previous literature. So is it likely? Yeah, you could say it's likely, it's plausible, but we don't have direct proof based off of this study. So just to wrap this up, the paper essentially shows us that water fasting leads to significant weight loss and fat loss. Not shocked at all. Water fasting changes the fat tissue's gene expression. We did see that, not after 1 day, but after 10 days. The gene signatures are from a lysosomal pathway, so creating these vesicles, and kind of autophagy related. So is that lysosomal pathway specific to autophagy, or is it being used for that fat breakdown? Well, probably both, to be honest. But again, we don't have specific proof of that. The macrophages have increased their gene expression of this spice gene, so you get more of this conversion of the monocytes to the macrophages with fat exposure. So they're trying to make that link, and I probably should have said this in more clearly before. They're trying to establish the link of, if the macrophages are exposed to fat molecules, what happens? Do you see more of these this gene expression that leads to these macrophages, from monocytes to macrophages, having them migrate from the bloodstream to the actual tissue? And the answer was yes, but only with the unsaturated fat, which is really, really interesting. So, the body does produce saturated fat, and it does produce unsaturated fat. So why would you have a stronger effect with unsaturated fat? I actually don't know the answer to that. So if you have any ideas, I'd be happy to hear them. Hopefully we'll discover that as we learn more. There is an increase in inflammatory signaling, but not all pro-inflammatory signaling with water fasting. And macrophages do invade the fat tissue with this added water fasting, with these 10 days of water fasting. And this was a really strictly controlled study, which was really great to see. So the big takeaway is this study, what can we actually take away from this? Like definitively, this study shows that water fasting leads to significant weight loss and fat loss and changes in gene expression. We know that for sure. And that may affect immune cells to invade fat tissue. Most likely that's the case, but we don't know if it induces them to take up these fat molecules that are then released by the fat cells. So that last part is not proven. It's really just kind of speculation based off of previous literature, trying to piece things together. So it opens up a lot of questions. It answers some things, and it offers some really intriguing effects that we can kind of get excited about, and hopefully we'll get more information out, because that would be really cool to know that there's this other mechanism for fat loss, that it's not just mediated by lipolysis, that the fat cells can, in certain situations, be so dumping fat so quickly, presumably, that they don't rely exclusively on lipolysis, through those three enzymes that are breaking apart these fat molecules, these more complex fat molecules, into simpler fat molecules. But they end up saying, you know what, screw it, we're just going to throw these fat molecules into these vesicles and just kick them out. And whatever can deal with it, that's what'll deal with it. And then these macrophages come crawling into that area and say, okay, well, I guess we're going to deal with this. We're going to be the ones to deal with this. So then maybe they suck it up, and then they break it down, and they continue to recruit more macrophages to go into that area to then deal with this excess fat release. So really, really interesting. Adds a lot of questions, but adds some really interesting hypotheses as well. So hopefully you found this informative. And with that said, I hope to have the pleasure of speaking with you in the next one. Have a good one, guys. See ya.